This is the first of 3 lectures on how cells harvest energy. Specifically, this is part 1 of 3, just an overview. So life is driven by energy. All of the activities that organisms carry out use energy. Here we will discuss the process all cells use to derive chemical energy from organic molecules and to convert that energy to ATP. Here I'd like to review the trophic classification that you learned about in Bio 117. Remember, some organisms are able to harvest the energy of sunlight through photosynthesis, converting that radiant energy into chemical energy. Those organisms are called autotrophs. They produce their own organic molecules through photosynthesis. The stored energy in the autotrophs is utilized by heterotrophs. Heterotrophs live on organic compounds produced by other organisms and stored in their tissues. Here, the autotrophs are the vegetation or plants. The rabbit is a heterotroph, taking advantage of the stored energy in the plant tissue. And the fox is also a heterotroph, taking advantage of the stored energy in the rabbit. Photosynthesis is the process by which autotrophs synthesize chemical bond energy using sunlight. The equation for that is carbon dioxide from the air plus water will yield glucose and oxygen. Cellular respiration is the process by which heterotrophs extract that energy from stored chemical bonds in order to make ATP. The equation for cellular respiration is glucose plus oxygen will yield carbon dioxide and water. Notice that these equations are the exact opposite. Do you think that the following statement is true or false? Only plants perform photosynthesis. Only animals perform cellular respiration. Hopefully you said false. Animals do not perform photosynthesis, that part is true, but plants do perform cellular respiration. After plants create glucose in the process of photosynthesis, they must then extract the chemical energy from the glucose to make ATP, because all organisms need ATP to actually do work. So plants do perform cellular respiration. This and the next few lectures will focus on cellular respiration. So here is the equation for cellular respiration in more detail. The reactants in the reaction are organic compounds, such as glucose, which is C6H12O6, plus oxygen, or O2, will yield carbon dioxide, CO2, plus water, which is H2O, plus energy in the form of ATP, and some of that energy will be lost as heat. Here again is the equation for cellular respiration. Glucose plus oxygen will yield carbon dioxide plus water. Some interesting questions here. Where does the glucose come from, and where does the oxygen come from? So where does the glucose come from? Well, of course it comes from your food. Most foods contain a variety of carbohydrates, proteins, and fats, all rich in energy-laden chemical bonds. Carbohydrates and fats, as you recall from previous lectures, possess many carbon-hydrogen bonds, as well as many carbon-oxygen bonds. We extract energy from all of the organic molecules we consume. In the case of the equation for cellular respiration, glucose is simply the representative molecule that we use. The end of this lecture series, part 3, will talk about the breakdown of the other types of organic molecules. The other question I address is, where does the oxygen come from? Well, of course, that comes from your breathing. Every cell in your body carries out cellular respiration, and so every cell in your body needs a constant supply of oxygen and molecules such as glucose. Here shows you how oxygen is delivered to each and every cell in your body. Your respiratory system takes in oxygen by breathing. Oxygen is inhaled by the lungs. It is delivered by the circulatory system to each cell in the body where cellular respiration is carried out. Carbon dioxide is given off as a byproduct of cellular respiration, and because that is a waste product, we need to get rid of it. So every cell in your body will offload the carbon dioxide into the blood, which will deliver it back to the lungs, where you will ultimately exhale it. Cellular respiration banks energy in ATP. Remember, we discussed in a previous lecture that ATP is the energy currency of the cell. ATP is needed to do the work. So in the process of cellular respiration, about 40% of the energy in glucose is actually converted into ATP energy. That's not very efficient. 60% of the energy in glucose is lost as heat. A muscle cell spends and reinvests about 10 million ATP per second. So I ask you, why is sweating necessary during vigorous exercise? Well, the more active you are, the more recycling of ATP you need to do, which means you need to break down more glucose, which means more heat is released as a byproduct. Your body needs to offload this heat, and that is why sweating is necessary. Sweating is a way for the body to effectively cool. As water evaporates off the surface of your skin, it takes a lot of heat with it. Remember, we are trying to extract energy from glucose in a way that it can be harnessed and used to make ATP. In the bottom of your screen shows the relative efficiency of burning glucose. So in the first scenario, literally, they have burning glucose in a beaker. The energy released from glucose in that situation, and the energy here is released as heat and light only, is 100%. 100% of the energy in glucose is released as heat and light. That is not energy that can be used by organisms. In the middle scenario, you have energy released from glucose and banked in ATP as the process of cellular respiration occurs. So burning glucose in cellular respiration, we are able to extract about 40% of the energy into banking ATP. As a comparison, if we were to compare this to burning gasoline in an auto engine, the gasoline energy that is converted to movement is only about 25%. So this being said, your body is actually more efficient at harnessing the energy in glucose than your vehicle is at converting energy into movement. The job of extracting energy from the complex organic mixture of most foods is tackled in stages. First, enzymes break down the large molecules into smaller ones. This is a process called digestion. Then other enzymes dismantle these fragments a bit at a time, harvesting energy from carbon-hydrogen bonds and other chemical bonds as well. The reactions that break down these molecules share a common feature. They are oxidations. Remember, an oxidation is a loss of electrons. Energy metabolism is therefore concerned with redox reactions, and to understand the process, we must follow the fate of the electrons lost from the food molecules. These reactions are not the simple transfer of electrons, however. They are also dehydrogenations. That is, the electrons lost are accompanied by protons. So that is what is really lost, is a hydrogen atom, not just an electron. Oxidation-reduction reactions play a key role in the flow of energy through biological systems because the electrons that pass from one atom to another carry energy with them. The amount of energy an electron possesses depends on its orbital position, or energy level, and around the atom's nucleus. When this electron departs from one atom and moves to another in a redox reaction, the electron's energy is transferred with it. Nicotinamide adenosine dinucleotide, or NAD+, is an electron carrier. It is a cofactor that helps enzymes catalyze redox reactions involving an energy-rich substrate. NAD plus accepts 2 electrons and 1 proton to become NADH, and this is a reversible reaction. This image shows you how NADH is an electron shuttle during cellular respiration. NAD plus accepts a pair of electrons and a proton from catabolized or broken down macromolecules or food molecules and is reduced to NADH. This animation shows the reduction of NAD plus to form NADH. So in it, you can see the enzyme in orange is going to interact with the cofactor NAD plus, will have its own binding site, and then a food molecule will have its own binding site. The food molecule is the substrate, and it has its own active site. Here the food molecule could be anything, but it's just showing you 2 hydrogens attached. After the food molecule or substrate and the NAD plus bind to the enzyme, there is a transfer of 2 electrons and 1 proton from the food molecule to the NAD plus, converting it into NADH. After that, both the food molecule, what is left of it, and the NADH are released. Here is the diagram from your textbook, which shows exactly what the animation did on the previous slide. Again, 2 electrons and a proton are transferred to NAD+, with another proton donated to the solution. Molecules that gain electrons are said to be reduced, and ones that lose electrons are said to be oxidized. NAD+ oxidizes energy-rich molecules by acquiring their electrons, and then reduces other molecules by giving the electrons to them. NADH is the reduced form of NAD+. So, cellular respiration is a redox reaction. So again, the equation, glucose plus oxygen yields carbon dioxide plus water plus energy. Here, glucose loses hydrogens. In a sense, it's losing electrons and protons as well. It's a dehydrogenation. But glucose loses hydrogens to become carbon dioxide, and thus it is oxidized. Oxygen, on the other hand, gains those hydrogens to become water. Because it is gaining hydrogens, thus electrons and protons, oxygen is reduced. In overall cellular energy harvest, dozens of redox reactions take place. Many electron acceptors, including NAD+, are used. In the end, high energy electrons from initial chemical bonds from food have lost much of their energy. This energy is transferred to a final electron acceptor. In aerobic respiration, or respiration that utilizes oxygen, the final electron acceptor is oxygen. In the absence of oxygen, anaerobic respiration may be used, in which the final electron acceptor is an inorganic molecule, not oxygen. Or fermentation, in which the final electron acceptor is an organic molecule. We will focus on anaerobic respiration and fermentation a little bit later. For now, let's just focus on aerobic respiration, where oxygen is used. Large amounts of energy must be released in small steps rather than all at once. Glucose must be oxidized in a series of steps. Take a car, for example. The more energy released in a single step, the more energy is lost as heat, and the less can be channeled to more useful paths. The same amount of energy is released whether all of the gas in a vehicle explodes at once or burns in a series of small explosions inside the cylinders of the vehicle. By releasing energy a little at a time, more energy can be used for movement rather than heat. So instead of releasing a single explosive burst of energy, electrons fall to lower and lower energy levels in steps, releasing stored energy with each fall. Here's the diagram from your textbook, which shows the electrons from food as they fall to lower and lower energy levels in steps, releasing stored energy with each fall as they tumble to the lowest or most electronegative electron acceptor, oxygen. And along the way, the energy released is used for ATP synthesis. Here again, using the step analogy, cells tap energy from falling electrons as they pass from your food to the electron shuttle, NAD+ to become NADH, down what is called an electron transport chain, and finally to oxygen, the final electron acceptor. Again, little bits of energy will be extracted along the way, and that extracted energy will be used to make ATP. There are many types of electron carriers. Generally, they are soluble, membrane-bound, and move within the membrane. All carriers are easily oxidized and reduced. Some carry just electrons, while others carry electrons and protons. Again, we are focusing here on NAD+, which will acquire 2 electrons and 1 proton to become NADH. So NADH is an electron shuttle. The enzymes involved here are called dehydrogenases. They remove 2 hydrogens from glucose. Now remember, each hydrogen contains 1 electron and 1 proton. So if 2 hydrogens are removed from glucose, that is a total of 2 electrons and 2 protons removed. 2 electrons and 1 proton are delivered to NAD+, causing it to become NADH. The other proton is released as a hydrogen ion. Here again is the reduction of NAD+ to form NADH. In a previous lecture, I introduced ATP as the energy currency of the cell. Cells use ATP to power most of those activities that require work, and all of those activities require the expenditure of energy by ATP hydrolysis. Cells use ATP to drive endergonic reactions that would otherwise not occur spontaneously. Again, endergonic reactions are reactions that require an input of energy. So remember, ATP works as a spring. The very highly negatively charged phosphate groups that form the triphosphate group in the ATP molecule, those negatively charged groups repel each other, and so there are very high energy bonds holding those together. So when the third phosphate group is cleaved off, an immense amount of energy is released to drive these reactions. There are 2 mechanisms for the synthesis of ATP. The first is what's called substrate-level phosphorylation. This requires the transfer of a phosphate group directly to ADP to create ATP. In substrate-level phosphorylation, ATP is formed by transferring a phosphate group directly to ADP from a phosphate-bearing intermediate or substrate. You will see this in this animation. The other mechanism for the synthesis of ATP is called oxidative phosphorylation. In oxidative phosphorylation, ATP is synthesized by an enzyme called ATP synthase, using energy from a proton or hydrogen ion gradient. This will be discussed in more detail once we get into the details of cellular respiration.